In this lesson, we will review TEKS B.7C, identify and illustrate changes in DNA, and evaluate the significance of these changes. This video is part 1 of a two-part series for B.7C. Part 1 reviews gene mutations and their effects, and Part 2 reviews chromosomal mutations and their effects. Mutation means change. What is a DNA mutation? A DNA mutation is a change in the nucleotide sequence of an organism's DNA. DNA mutations can have a variety of causes, including environmental factors called mutagens. For example, the sun's rays can be a mutagen. Extended exposure to sunlight can cause mutations in the DNA of skin cells. DNA mutations can be categorized into two groups: chromosomal mutations and gene mutations. A chromosomal mutation affects large regions of DNA involving many genes and entire segments of a chromosome. A gene mutation affects a short sequence of DNA such that only one gene is involved. Let's take a closer look at both categories of mutations, starting with gene mutations. A gene mutation is a change in the DNA nucleotide sequence of one gene. There are three types of gene mutations: deletions, insertions, and substitutions. Let's take a closer look at each of these, starting with deletions. A deletion is a mutation in which one or more base pairs are deleted from a DNA nucleotide sequence. Let's watch an animation of a deletion. This model shows the two strands of a DNA molecule. Let's watch as a CG base pair is deleted from the DNA nucleotide sequence. Notice that all the other base pairs in the sequence remain unchanged. Next, let's take a closer look at insertions. An insertion is a mutation in which one or more unintended base pairs are inserted into a DNA nucleotide sequence. Let's watch an animation of an insertion. In this segment of DNA, a CG base pair is being inserted into the nucleotide sequence. All of the other base pairs in the sequence remain unchanged. Next, let's take a closer look at substitutions. A substitution is a mutation in which one base pair in a DNA sequence is replaced with a different base pair. Let's watch an animation of a substitution. In this segment of DNA, a TA base pair is being replaced with an AT base pair. All of the other base pairs in the sequence remain unchanged. To review, a deletion is a mutation in which one or more base pairs are deleted from a DNA sequence. An insertion is a mutation in which one or more unintended base pairs are inserted into a DNA sequence. And a substitution is a mutation in which one base pair in a DNA sequence is replaced with a different base pair. How do mutations impact the cell? A gene mutation can result in errors in protein synthesis. Remember that DNA serves as the blueprint for building proteins. Cells rely on proteins to carry out life processes. So, how does the mutation of an individual gene impact a protein? To answer this question, we must review the role that genes play in protein synthesis. A gene's nucleotide sequence codes for a complementary strand of mRNA. The mRNA strand's nucleotide sequence codes for a specific sequence of amino acids. The amino acids form a polypeptide chain, and the polypeptide chain forms a protein. This illustration shows a polypeptide chain folding into a protein. Why is the specific sequence of amino acids in a polypeptide chain important? It impacts the protein's shape and size. Now, think about how a gene mutation might affect this process. Look at the model of the gene. A base pair is being highlighted. If a gene's nucleotide sequence were changed due to a mutation, its mRNA transcript would be incorrect, which means the resulting amino acid sequence will likely be incorrect as well. In this way, gene mutations can cause errors in protein synthesis. Now, let's look at the specific kinds of protein synthesis errors that gene mutations can cause. Gene mutations can cause 4 kinds of protein synthesis errors: missense mutations, silent mutations, nonsense mutations, and frame shift mutations. Let's look at examples of each of these, starting with missense mutations. In this animation, we can see a template DNA strand, its mRNA transcript, and the resulting amino acid sequence. Remember that during protein synthesis, the ribosome reads mRNA in the form of triplet code. Each segment of 3 RNA nucleotides calls for a particular amino acid. Look at the 4th mRNA triplet, GGC. GGC codes for the amino acid glycine. Now, let's watch what would happen if the template DNA triplet underwent a mutation. The first base, cytosine, is substituted with thymine. As a result, its complementary base in the mRNA transcript will be adenine instead of guanine. The RNA triplet will now read AGC, which codes for the amino acid serine. So, the substitution mutation will result in an incorrect amino acid, serine, being placed in this polypeptide chain. To summarize, we can say that missense mutations result in exactly 1 incorrect amino acid being placed in a polypeptide chain, and they are caused by substitutions. Now, let's look at silent mutations. In this animation, we can see a template DNA strand, its mRNA transcript, and the resulting amino acid sequence. Let's watch what would happen if the template DNA strand underwent a substitution mutation. The 6th base in the strand, cytosine, is substituted with thymine. As a result, its complementary base in the mRNA transcript will be adenine instead of guanine. So, the RNA triplet now reads AAA instead of AAG. Fortunately, AAA codes for lysine just as AAG did. So, the substitution mutation will not impact the polypeptide chain. The mutation is silent. Next, let's look at nonsense mutations. In this animation, we can see a template DNA strand, its mRNA transcript, and the resulting amino acid sequence. Let's watch what would happen if the template DNA strand underwent a substitution mutation. The 4th base in the strand, thymine, is substituted with adenine. As a result, its complementary base in the mRNA transcript will be uracil instead of adenine. So, the RNA triplet now reads UAG instead of AAG. AAG codes for the amino acid lysine. UAG, on the other hand, is a stop codon. It does not code for an amino acid. It signals to the ribosome that the polypeptide chain is complete. Hence, a stop codon stops protein synthesis. The result is significant. This polypeptide chain will be incomplete. It will be shorter than its intended length and most likely unusable. Next, let's look at frameshift mutations. In this animation, we can see a template DNA strand and its mRNA transcript. As we know, each of these codons translates to an amino acid as shown, and this codon happens to be a stop codon. Now, what would happen if the template DNA strand had a deletion mutation? Let's watch as the 9th base in the template strand, adenine, is deleted. As a result, the mRNA transcript will be missing a base as well. This mutation is going to affect multiple amino acids and the stop codon. To explain this, let's read the triplet code, one codon at a time. The first triplet hasn't changed at all, so it will still code for the amino acid methionine. The next triplet hasn't changed either, so it will still code for lysine. The next triplet is the one with the deletion. Because a base is missing, the cell has no choice but to read this RNA triplet as UUG. UUG will code for the amino acid leucine instead of the phenylalanine that was intended. And that's just the first error. The next RNA triplet now reads GCU, which codes for alanine. But it was supposed to be glycine here. And as you may remember, the next triplet was supposed to be a stop codon, but now it codes for the amino acid lysine. All of this was caused by a single deletion mutation. One missing RNA base caused the entire reading frame to be shifted, resulting in a potentially lengthy and damaging series of errors in the polypeptide chain, hence the term frameshift mutation. Note that just like deletions, insertions can also cause frameshift mutations. These 4 types of mutations, missense mutations, silent mutations, nonsense mutations, and frameshift mutations, demonstrate how gene mutations impact protein synthesis. Now, how do these changes affect an entire organism? Gene mutations can have a variety of effects on organisms and populations. Most instances of DNA damage are neither harmful nor beneficial. Why is that? One reason is that most of the time, DNA damage is repaired before it can cause any harm. For example, cells have sophisticated mechanisms for immediately repairing DNA errors that occur during the process of replication. But what if DNA damage doesn't get repaired? Remember that some gene mutations are silent. As we learned earlier, silent mutations do not impact protein structure in any way. And lastly, even when protein structure is impacted by a mutation, sometimes the structural issue is minor enough that the protein can still perform its function. Some gene mutations can be harmful to organisms. They can cause genetic disorders that have a significant impact on an organism's functioning. For example, in humans, gene mutations can cause colorblindness, sickle cell disease, and cancer. Earlier, we reviewed substitution mutations. Did you know that sickle cell disease is caused by a substitution mutation? Let's watch how this happens. Sickle cell disease affects the shape of red blood cells. This is a normal red blood cell. Notice its round shape. And this is a sickle cell, shaped like a sickle. The abnormal shape of the sickle cells is caused by an abnormality in the protein hemoglobin. Here we see the normal molecular shape of hemoglobin. And here we see the abnormal shape of hemoglobin in sickle cells. So, what causes this abnormal shape of hemoglobin? You're looking at part of a hemoglobin gene. Here is the DNA, and here is its mRNA transcript. A normal hemoglobin gene contains this codon, which codes for the amino acid glutamine as shown. But in individuals with sickle cell disease, this codon has a mutation, which we're about to watch. The substitution of a T with an A in the DNA causes a domino effect. The mutated codon now codes for valine, which is the wrong amino acid. This seemingly simple mutation changes the entire shape of the hemoglobin protein, resulting in sickle cells. This is just one of many examples of a small change in DNA leading to a disease. We've discussed neutral and harmful gene mutations. Gene mutations can also be helpful to organisms and populations. Occasionally, a mutated gene can result in an advantageous trait, a trait that helps an organism thrive in its environment. Also, gene mutations are an important source of genetic variation within populations. The trait variations produced by mutations can help a population withstand environmental changes over time. Note that in sexually reproducing organisms, for a mutation to be passed from parents to offspring, the mutation must be present in the gametes. Now you know how gene mutations can have neutral, harmful, and helpful effects on organisms and populations. Let's review the vocabulary of this lesson. A gene is a segment of DNA that influences a particular trait. A gene carries genetic instructions in its nucleotide sequence. A gene mutation is a change in the DNA nucleotide sequence of one gene. A deletion is a gene mutation in which one or more base pairs are deleted from a DNA nucleotide sequence. An insertion is a gene mutation in which one or more unintended base pairs are inserted into a DNA nucleotide sequence. A substitution mutation is a gene mutation in which one base pair in a DNA sequence is replaced with a different base pair. An amino acid is a building block molecule of a protein. An amino acid links with other amino acids to form a polypeptide chain. A polypeptide chain is the end product of translation. It is a chain of amino acids, and it forms a protein. A codon is a 3-nucleotide sequence that is read during translation. A frameshift mutation is a gene mutation that causes a shifted reading frame during translation, resulting in a series of errors in polypeptide synthesis. A missense mutation is a gene mutation that results in exactly one incorrect amino acid being placed in a polypeptide chain. A nonsense mutation is a gene mutation that produces an unintended stop codon, resulting in an incomplete polypeptide chain. A silent mutation is a gene mutation that does not impact protein synthesis because the mutated codon still codes for the appropriate amino acid. A mutagen is an environmental factor that can cause a DNA mutation. Tobacco smoke and the sun's ultraviolet rays are two of many examples of mutagens. In this lesson, we reviewed TEKS B.7C, identify and illustrate changes in DNA and evaluate the significance of these changes. This video is part 1 of a 2-part series for TEKS B.7C. Part 2 reviews chromosomal mutations and their effects.